Hi everyone. The video is on prokaryotic DNA replication. Now what is DNA replication? It is nothing but the synthesis of new DNA. So two dotted DNAs are formed from a double-stranded parental DNA. Now how does this happen? The DNA first unwinds itself and each of the strand of the DNA acts as a template to which a complementary strand is synthesized. So that is why even you call this as semi-conservative replication, because the parental strand in the daughter DNAs, the parental strand is, one parental strand is intact, yes, or the parental strand is conserved in each of the daughter DNAs. So that is why you call it as a semi-conservative replication. And what are the requirements of a DNA replication to take place? First of all, DNA strand, of course, and then DNA helicase enzyme, single-stranded binding proteins, DNA topoisomerase, magnesium-dependent DNA polymerase, DNA primase for synthesis of RNA primers, and DNA ligase. So these are the enzymes and proteins which are required for the DNA replication. Now, what is the first step? For the DNA replication to take place, first the replication origin has to be identified. So one site has to be identified. So that site is nothing but called the ORI. So origin of replication or the ORI is first identified. So how is it identified? It is a short AT rich segment. Yes, it is a short AT rich segment. Usually AT rich segments are the first origin of replication. Yes, because AT is easier to break than the GC, because AT has 2 hydrogen bonds and GC has 3 hydrogen bonds. So first, as we have to unwind the parental strand, parental DNA, the origin of replication or the ORI is AT rich segment. So that is first identified. So in a prokaryote there is a single ORI, but in a eukaryote, yes, in a eukaryote there can be multiple sites of origin of replication. Yes. So as you find the origin of replication, initially the unwinding of that area happens. So as unwinding of that area happens, it looks like a bubble. So this is nothing but the formation of replication bubble. So the replication bubble, as it happens, now the DNA synthesis can take place in both the directions. So since it is a prokaryote, there is a circular DNA. Yes. So the whole of the circular DNA has to be replicated now. So as we have found the origin of replication at one site, from both the sides, yes, in both the directions, the DNA has to be replicated. Yes, and finally what do we get? We get two daughter DNAs. So this happens in the prokaryotes with a single origin of replication. Similarly, there are multiple origin of replication in a eukaryote. So in each of the replication bubble, alongside each of the replication bubble, in both the directions the synthesis happens. Finally, we get two double-stranded DNAs. Yes. The next step is unwinding of the double-stranded DNA. So as we have found the origin of replication, now we have to unwind the double-stranded DNA. Unwinding is done with the help of the enzyme DNA helicase. So what is the enzyme which is helping in the unwinding of DNA strand? It is nothing but the DNA helicase. Now as it binds to the origin of replication, it starts unwinding. So it starts separating the two strands of the DNA, and this is an ATP dependent process. Now we all know the DNA, the two strands of the DNA are complementary to each other. So even if you separate, it has a higher tendency to reattach. Yes. So to prevent this attachment of this two strands of the DNA, there are single-stranded binding proteins which bind to the two strands of the DNA as and when the helicase enzyme unwinds the DNA, right? So what have we seen now? The DNA helicase, which helps in the unwinding, and single-stranded binding proteins which keeps the strands separated. Yes, so these are the two Single-stranded binding proteins and the helicase are the two requirements for unwinding of the DNA strand and formation of replication fork. So this is the replication fork. You can appreciate this, right? This is the replication fork. So the DNA helicase keeps unwinding the DNA in this direction, and single-stranded binding proteins bind to the two strands of the DNA and keeps it separated. Yes. The next step is, as you keep unwinding, as you unwind the DNA, what happens? There are supercoils which can be formed. So the supercoils can, the positive supercoils can, or the negative supercoils which can be formed. So as the DNA gets unwinded, what happens? Look at this, there is torsion here. So thus the positive supercoils are occurring ahead of this strand separation. Yes, so so how should we release these supercoils? We have to release these supercoils, right? So the enzyme which helps in releasing these supercoils are DNA topoisomerases. So there is DNA topoisomerase 1 and DNA topoisomerase 2. So there are two DNA topoisomerases, type 1 and type 2. Now type 2 is ATP dependent and type 1 is non-ATP dependent. It's not ATP dependent. Yes. So how does the type 1 DNA topoisomerase act? It creates a nick in one strand of the DNA. It creates a nick and clears the supercoils, and again the nick is sealed. Okay. So along the one strand of DNA a nick is made, and then as the supercoils get corrected or released, the nick is sealed. So this is nick is sealed by this DNA topoisomerase 1. This is the action of type 1. Now type 2, what is happening? Type 2, this is a circular DNA which is found in the prokaryotes, right? So what happens? This is now, there is a positive supercoil. Now it forms nick along two strands of the DNA. So in both the strands of the DNA, nick is made and a negative supercoil is created. So when a positive supercoil, now look at this, when a positive supercoil occurs, this negative supercoil can relieve those positive supercoils. Yes, are you able to understand? So this is what is done by the type 2 topoisomerase. So what does this do? It is able to create a negative supercoil in the circular DNA using energy. Of course, it is using this ATP from the hydrolysis of ATP, right? So this facilitates the replication of DNA because the negative supercoils will neutralize the positive supercoils which will be introduced while unwinding the double helix. Yes. So these are the actions of DNA topoisomerase 1 and 2 in releasing the supercoils. So now what have we done? We have identified the origin of replication. We have formed the replication fork. So there is unwinding of the DNA strand using DNA helix and single-stranded binding proteins. And then whatever supercoils has been created, we have released those supercoils using DNA topoisomerase. Now we have to understand the two concepts. One is the lagging strand, the other one is the leading strand. So what are they? Here when you see, this is the origin of the circular DNA we have seen, right? This is the circular DNA, and we have identified one. So as the origin of replication is this, yes, is this. Now identify this strand is in 3' 5' direction. Yes. So always remember, the synthesis of DNA can occur only in the 5', 3' direction. The synthesis can occur only in the 5', 3' direction. Yes. Now this parental strand is in the 3', 5' direction. Yes, are you able to appreciate? So synthesis in the 5', 3' direction is in this direction. This is the 5', 3' direction. So that is towards the replication fork. So as and when the replication fork opens, new nucleotides can be added and DNA can be synthesized in a continuous manner. Yes, do you agree? So the strand which is synthesized continuously towards the replication fork in the 5' 3' direction is called the leading strand. Now whereas look at the other end, this is the replication fork. Yes, this is the replication fork. Now here what happens, this is the 3' and this is the 5' end. Yes, this is the 5' end. So the 5' 3' direction is in this direction, which is away from the replication fork. Now imagine from here a new DNA has been synthesized. Again the DNA helicase will unwind this DNA. Yes, in this direction. So as and when it unwinds, we have to synthesize a new DNA. So we can do it in the 5' 3' direction in a discontinuous manner only. Yes, in a discontinuous manner only. So consider this is the replication fork. This is the replication fork. Yes. Now the DNA helicase keeps unwinding the DNA in this direction. Now I have synthesized a new strand along this direction. So this is my 5' 3' direction. So I have synthesized a new DNA strand complementary to this parental strand in this direction. Now the DNA helicase again unwinds. Yes, it again unwinds. So what happens? My fork is here. Yes, so again I have a new area which has to be synthesized. So again along this direction a new DNA will be synthesized, but in a fragment wise. So the DNA has to be synthesized discontinuously as fragments as and when the DNA helicase opens this parental strand. So this strand which is synthesized discontinuously, yes, discontinuously away from the replication fork, is called the lagging strand. So the strand which is synthesized continuously is the leading strand. The strand which is synthesized discontinuously away from the fork is the lagging strand. Now we have covered this one part only. Yes, one part. So what have we synthesized? Along this circular DNA, one part is what we have seen. We have the other part also, right? So the synthesis has to be taken place on the other side also. So what will happen for the other side? What happens? The replication fork is on this side. So towards the replication fork, if the synthesis can happen in the continuous manner, so that becomes the leading strand. And when the synthesis of DNA is away from the replication fork, it happens in fragment wise or discontinuously, so that becomes the lagging strand. So don't get confused with this diagram. So on one side it is a leading strand, on the other side it will be the lagging strand, because the replication fork is on this on both the ends. So hope you understand this picture. So this is the leading strand and the lagging strand. Now how is the DNA synthesized? So the new DNA has to be synthesized using the enzyme DNA polymerase. Okay, but DNA polymerase has the capability of attaching nucleotides to the strand, DNA strand, but it cannot happen from the scratch. So what you have to do, you have to attach one RNA primer. RNA primer is nothing but a short piece of or short fragment of nucleotides, which is single stranded RNA, which is a short piece of nucleotide. We have to attach this RNA primer to one end. Yes, you have to attach the RNA primer to one end. so that DNA polymerase can attach nucleotides to the starting from the end of this RNA primer. Yes, so this is the RNA primer. To this RNA primer, the DNA polymerase can attach nucleotides. So as triphosphates, as deoxynucleotide triphosphates, these nucleotides will come and attach. So these deoxynucleotide triphosphates, as it gets attached to the RNA primer, to one end of the RNA primer, two phosphates are released. So a monophosphate is what is getting attached. So this is the RNA primer. To the RNA primer, one deoxynucleotide has been attached. Yes, so a new 3' 5' phosphodiester bond is being created and one nucleotide is being attached to this RNA primer. Further to this nucleotide, with the help of DNA polymerase, the further nucleotides can be attached, and these nucleotides, how can they be attached? These nucleotides should be complementary to the parental DNA or the parental template. Yes, these nucleotides are chosen in such a way, chosen by the DNA polymerase in such a way that they are complementary to the parental template. So which initiates replication? So DNA polymerase cannot add nucleotide as such. So it needs an RNA primer. So an RNA primer is what is attached first, after which a DNA polymerase can attach the nucleotides. Yes, so this is how the initiation of the DNA synthesis happens. So look at this picture. If you see this picture, you'll be able to understand the whole thing. This is the leading strand and this is the lagging strand. Yes. So why do you say it is the leading strand? See, this area is the replication fork, or this area is where the DNA helicase is there. So DNA helicase will start unwinding the parental DNA, and the single stranded binding proteins will help separating these DNA strands. So as the unwinding happens, there are supercoils which can be created. So you have to release these supercoils with the help of enzyme topoisomerase. After that, along the leading strand, so this is along the leading strand, the direction of synthesis is along the 5' 3' direction. So for the newly synthesized DNA, this is the 5' 3' direction. So since it is the 5' 3' direction, so as and when the helicase unwinds the DNA, the synthesis can happen continuously. Yes, so this is with the help of initially one RNA primer is attached. Yes, initially one RNA primer is attached, after which DNA polymerase can help in forming these addition of and adding these nucleotides. Thus a newly synthesized DNA is formed in a continuous manner along the lagging strand. So which polymerase is helping in this? DNA polymerase 3. DNA polymerase 3 is what recognizes the RNA primer and it will begin to synthesize the DNA. Yes. Similarly, what happens in the other side? In the other side, this direction is the 5' 3', this direction is the 5' 3' direction. Yes, so you have to synthesize the DNA along this 5' 3' direction. So initially the DNA helicase was here. Okay, initially the DNA helicase was here. It was unwinding the DNA. So the RNA primer comes and sits here. After the RNA primer recognition, the DNA polymerase has started to synthesize the DNA in this direction. Yes, now what happens? This DNA helicase now starts unwinding the DNA and it has moved to this area, So again what happens? An RNA primer comes and sits here. Then DNA polymerase recognizes this and it will start synthesizing the DNA along the 5' 3' direction. So there is a gap here. Yes, there is a gap here. There is a gap, of course, right? And another RNA primers is there. So you need so many RNA primers along the lagging strand to synthesize DNA. So I have now synthesized the DNA in fragments. So this is one fragment, this is one fragment. These fragments are called Okazaki fragment. These fragments are called Okazaki fragments. Yes. Finally, I have to join these fragments to form one single DNA. One single strand. Yes, I have to join these fragments. So how do I join these fragments? Using DNA, I will tell you, using DNA polymerase 1 and DNA ligase, I am going to join those fragments. Yes, so look at this picture, DNA polymerase 1. What is the action of DNA polymerase 1? There are so many RNA primers. So as and when DNA helicase moves to unwind the double-stranded DNA, there are numerous RNA primers along which the DNA polymerase can synthesize in the 5' 3' direction. Yes, so now I have these many fragments called Okazaki fragments along the lagging strand. Now what happens? DNA polymerase 1, the DNA polymerase 1 now removes these RNA primers. DNA polymerase 1 removes these RNA primers. Also, DNA polymerase 1 fills the gaps which are present. Yes, fills the gaps which are present. So, DNA polymerase 1 can remove these RNA primers and it also fills the gaps which are present. So, after which what happens? There is the DNA ligase which can seal the remaining gaps. Yes, so there are numerous fragments which are formed. In between the fragments, I have got the primers. So RNA primer has started the elongation of these DNA segments, right? So these Okazaki fragments I have to join together. So using DNA ligase, I am going to remove these RNA primers and then I am going to seal it. Yes, I am going to seal these Okazaki fragments using DNA polymerase 1. DNA polymerase 1 adds nucleotides along the gaps. So adds nucleotides along the gap and DNA ligase will form phosphodiester bonds and seals the nicks present. So thus it forms one continuous segment, one continuous strand. So this is the action of the DNA polymerase 1. Now what is the function of DNA ligase? So as I had said, this is the parental strand. This is the Okazaki fragments. Yes, this is the lagging strand. So we have a gap here. We have a gap. So what are we doing? We are forming a phosphodiester bond using the DNA ligase enzyme. Yes. So this is the function of DNA ligase. Now what is the function of DNA polymerase 3? One function we know that it adds the nucleotides. Yes. So this needs the RNA primers. This need RNA primer. After an RNA primer, following this RNA primer, the DNA polymerase can add nucleotides. It chooses the nucleotides in such a way that they are complementary to the parental strand. Now, it has the 5' 3' polymerase action. That is, along the 5' 3' direction, it adds nucleotides. Yes. So it adds nucleotides which is complementary to the parental strand. So this is the parental strand and this one is the newly synthesized strand. So newly synthesized strand will be complementary to the parental strand. So this activity is nothing but 5' 3' polymerase activity. Now it also has another reaction which is the proofreading action. Proofreading action is nothing but proofreading action is along 3' 5' exonuclease activity. Yes, 3 prime 5 prime exonuclease activity. Now that we have added one nucleotide, after adding the nucleotide, this DNA polymerase will recheck whether the added nucleotide is complementary to the parental strand. If it is not complementary to the parental strand, then we have to remove it, right? So removing is done in the 3 prime 5 prime. It is going back and removing the nucleotide. Yes, so that is why it is 3 prime 5 prime exonuclease activity. So thus the nucleotide which is added, which is not complementary to the parental strand, is removed from the strand. So this is also done by the 3 prime 5 prime exonuclease activity of the DNA polymerase 3. So how many functions does DNA polymerase 3 has? It has 5 prime 3 prime polymerase activity and 3 prime 5 prime exonuclease activity. These are the two functions of DNA polymerase 3. So now that what we have seen initially the origin of replication, finding out the origin of replication. Aura is nothing but a short segment of 80 rich nucleotides. Okay. After that, the strand is there is unwinding of the DNA strand using DNA helicase, and the single stranded binding proteins will keep the strands in a separated condition. So replication fork is formed. So as the DNA helicase unwinds the DNA strand, there are coils, supercoils formed. Those supercoils are released using DNA topoisomerase enzymes 1 and 2. Yes, and there is along the replication fork, in one end the DNA synthesis can be taken place in the 5 prime 3 prime direction towards the replication fork continuously. That strand is called the leading strand. And in the other side, the DNA fragment has to be synthesized away from the replication fork. So it can happen in a discontinuous manner. So that is the lagging strand. So for synthesizing, for initiation of the synthesis of DNA, an RNA primer is needed. Okay, an RNA primer is needed. And what gives the RNA primer? The enzyme primase. Okay, primase, this is an RNA polymerase. So the primase enzyme forms the RNA primers. So from the RNA primer is being recognized using DNA polymerase 3, after which it adds the nucleotide. So as triphosphates get added after releasing two phosphate groups. So a monophosphate will be attached along the DNA strand. And further addition of nucleotides will happen depending upon the parental strand nucleotides. Now after that, DNA elongation happens. So along the lagging strand, it happens in discontinuous manner. So what are those fragments? They are Okazaki fragments which are formed in a discontinuous manner along the lagging strand. So there are numerous RNA primers which are needed to synthesize strand in the lagging strand, whereas only one RNA primer is necessary for synthesis for the DNA synthesis along the leading strand. Then DNA polymerase 1 will recognize these RNA primers and it will remove these RNA primers. After that, the gaps are being filled by these DNA polymerase 1 by addition of nucleotides. After the nucleotides are added, DNA ligase will form phosphodiester bonds. Yes, so thus two continuous strands can be formed. Now, the action of DNA polymerase 3 is, there are two actions. One is the 5 prime 3 prime polymerase action and 3 prime 5 prime exonuclease action, which is helping in the proofreading of the DNA. So this is the DNA replication steps which is happening in the prokaryotes. Thank you.